Hey up everyone, welcome to this PowerPoint presentation on how to answer the first question for the scene test of Introduction to Cell Biology. What we're going to do today is learn together the content for the question, and then hopefully by the end of it we can check your learning and then apply it and go on to being successful in the test. So what we're going to do is learn everything that you need to know to answer that first question. And the intentions of this session are, first of all, to explain the difference between active and passive transport processes. So that's the first thing we need to explain, and it's the first thing you need to explain when you answer the question in the test. The second thing that we're going to do is identify and explain 3 passive and 3 active transport processes. So there are 3 active methods, there are 3 passive methods, and in order for you to show that excellent understanding that I'm looking for in the test, you're going to be able to draw a diagram of each and also identify the type of molecule that uses each method. So essentially there's an introduction, so what is active and passive transport, and then you need to go into 3 active and 3 passive transport methods. Now that will mean some kind of diagram with some kind of written explanation about what's going on, and then an example of a particular type of molecule, and that will nail the criteria, and you really can't do any more than that. So without any further ado, let's get into it. Okay, before we talk about what active and passive processes are, it's first of all important to understand that cells are a fundamental unit of life. We're made of them, and there's constant traffic going in and out of the cell. We need nutrients to keep this thing alive. We also need to get rid of waste products so that we can get rid of them. We also, this cell could be exporting important products to the rest of the body, such as hormones. And so there is constant traffic across the cell membrane. Now, if the cell does not need any energy to do this, then they are called passive processes. So a cell can move these things in and out across the cell membrane, across the plasma membrane, without using any energy at all, and they happen passively. And this is what is summarized in the key points at the bottom. Now, if you're wondering what that is, ATP, that is just another term for energy. So a cell does not need to use ATP to move these substances across the membrane, and all ATP means is energy. So very simply, active processes, as you can see down in the key points section, explains that active processes requires energy or ATP required from the cell to move the molecules. So the cell needs to invest some of this hard-earned energy that we've required to move these processes. So often they're against the concentration gradients or doing really complex things. And I like to think of it as, you know, almost going up the hill. So we tend to go from an area of low to high, because if we imagine that boulder, you've got to really put some effort in to going up that hill or pushing that that boulder up the hill. So active processes are ones that the cell needs energy to do those processes. Very simply. As you can see from the graphic, there are, and as I've already said, there are 3 passive processes and 3 active processes. We're going to talk about the passive ones first, and then we'll talk about the active ones. So the first one, obviously, as we can see, is passive diffusion. The second one will be facilitated diffusion, and the third one would be osmosis. The fourth one we'll talk about, or the first active process, would be a protein pump, endocytosis, and then exocytosis. So 6 processes. You need to be able to draw a diagram of each, explain what's going on, maybe bullet points to explain, and then an example of a molecule that is moved by each of the process. So the first of our passive processes that we're going to learn about is passive diffusion. And as the name suggests, passive diffusion is a passive process, which means that no energy is needed to move these molecules using this method. Now, the first concept that we need to sort of concern ourselves with is the concept of high concentration and low concentration. So if we start by putting some green molecules over here, and we put a few over here, then the area of high concentration would be up here. And that means if we sort of took a square of this size and we took a square of this size, the same size should I say, and we looked at how many molecules were in that little square, and that could be anywhere, you could make a square above it or to the right of it, we would know that there's more molecules here than there is here. So this would be the area of high concentration, where I've put the H. This would be the area of low concentration, and same here, this would be low and this would be low. Now, in science, molecules that have a certain degree of heat have a level of kinetic energy. That means that they move. So if you apply heat to something, it moves, just like if you're boiling water, it starts to move. Now, your bodies are at 37 degrees usually, and so they have some kind of heat, and those molecules will move. And so when they move, they will generally collide and bish and bash to areas of low concentration. So these molecules will spread out. They'll spread out to achieve relatively a random balance across the area. Okay, now that is passive diffusion. It's where molecules move from areas of high concentration to low concentration. Now, when we're talking about passive transport methods, we're talking really about a cell. So here is a cell, and obviously this is the inside of the cell, and this is the outside of the cell. Now, when we breathe in, for example, what we're taking in is a lot of oxygen. So if I draw a lot of oxygen in red out here, then what we can see is that the oxygen transports around the blood to our cells, and it produces an area of high concentration of oxygen outside of the cell. Obviously, inside of the cell, in this diagram, is a low concentration. And so what happens is this oxygen moves into the cell from high concentration to low concentration without using energy. It happens passively. Now, it's able to move across this phospholipid bilayer or cell membrane because the molecule is lipid soluble and small enough to fit through. Now, if you've not done the lesson on the structure of the cell membrane yet, then don't worry. All you need to know is that this black line is made of lipids, something called lipids, which is like a fat, a liquid fat. And so oxygen is lipid soluble. That means that it can dissolve in the lipid, it can dissolve in this black line and come out of the other side, and it's also small enough to do so. So what we get is a movement from high concentration to a movement of low concentration because the molecule is lipid soluble, i.e. dissolvable, and small enough to do so. Now the question is, why does oxygen come into the cell? Well, you may not know, but you're breathing in oxygen in order for you to create energy. And there's a whole unit on this, so I don't want to get too technical. So we take in oxygen into every one of our cells to create energy. Now, as a byproduct of that, we also create inside of the cell a waste product. And while I'm drawing these, you can have a think of what the waste product might be. And if you haven't already thought about it, the cell produces something called carbon dioxide, or CO2. So what we get is a buildup of carbon dioxide inside the cell. So we get a high concentration of carbon dioxide building up inside the cell. Now, lucky for us that carbon dioxide is also lipid soluble, and it's also small enough to fit through the plasma membrane or the cell membrane. So what we get is this movement of carbon dioxide out of the cell by passive diffusion. The cell does not use energy to do this. It happens magically. It happens wonderfully. So this carbon dioxide, which is a waste product, goes back into the blood and taken back to your lungs to be excreted or breathed out. And it's what you're doing right now, every day, every minute of every hour. You're breathing in, and that oxygen is coming into your blood from the lungs. That oxygen then is being deposited in every orifice of your body, in every cell of your body. That oxygen then diffuses into the cell, in which you create energy from that. And as a byproduct, you create the carbon dioxide, which comes out of the cell back to your lungs, and which you breathe out. So that is an example of passive diffusion. The key points here: it's a passive process. It doesn't need energy. It moves from areas of high concentration to low concentration directly across the cell membrane, and it does so because the molecules are lipid soluble and small enough. The examples being oxygen and carbon dioxide. Funnily enough, alcohol does also move across the cell membrane from passive diffusion, but the less we talk about that, the better. Right, so the second passive type of transport that we can talk about is something called facilitated diffusion. And facilitated diffusion, as it says in the key points, is still a passive process, and no energy is required from the cell. Now, it is a type of diffusion, as it says in the title, so there is still this movement from high concentration to low concentration. But this particular movement process is for molecules that are too large to fit across the cell membrane directly and are too lipid insoluble, which means they don't dissolve in the cell membrane, so they can't get across. So first of all, let's start by drawing a cell membrane, and I'm going to draw these two sort of squiggly circles, you can call them. Now these are my protein channels, so we've got one green and one blue. So let's say we have a build-up of this molecule outside of the cell membrane, and this molecule is too large and too lipid insoluble to get across the cell membrane. So if it has this random kinetic energy and it bumps into the cell membrane, it will be repelled by that phospholipid bilayer. So how does it get into the cell? Well, you can obviously guess, we've already said it, or it's in the key points. It uses special protein channels. So it goes from areas of high to low concentration using these protein channels. It bypasses the properties of the cell membrane so that these molecules can move from high to low. Now, alternatively, and just for an example, it can move the other way. So molecules can move from inside the cell to outside the cell, from areas still of high concentration to low concentration using these protein channels. Again, they're large, they're lipid insoluble, so if this green molecule here decides to hit this part of the cell membrane, it would be repelled because it's not using a protein channel. It has to use a protein channel. Now, we get a lot of health professionals and a lot of, I don't know, midwives, paramedics, physios thinking, why the hell do I need to learn about cell biology? What has this got to do with being a nurse or a paramedic? Well, these processes are underpinning disease and illness, and they underpin how the body works. So it's really important that you get a fundamental understanding of how this body works so that you can identify and deal with these issues when they come up. So for example, a molecule that moves by facilitated diffusion is glucose. So this blue molecule here, we can say is glucose. Now when somebody eats a meal, when you eat your breakfast for example, the sugars in your breakfast will be converted into glucose and absorbed in the blood. So let's draw a little blood vessel. Now this glucose travels around your body in your capillaries, arteries and veins, and this glucose then diffuses out into the fluid, into the surrounding areas of your cell, and then what happens is it gets absorbed into the cells. Okay, now in order to open these special protein channels, what the body does is release a hormone which you have probably heard about, called insulin. So as soon as you eat a meal, your body triggers the release of insulin from your pancreas, and it's this insulin that travels to these channels and opens them up so that the glucose then can get into the cell by facilitated diffusion. Why does the cell need glucose? Well, the cell needs glucose for energy. Without getting glucose in your cell, we become very inefficient at producing energy, and we have lots and lots of problems. One of the ways that we might not be able to get this glucose within a cell is a condition called diabetes. Now it's very early to be talking about this, and you will learn about this towards the end of the year in your final exam. But it just gives you an idea of why it's important to learn about this cell biology, because all of these things underpin the stuff that you're going to be learning later on. This is how reproduction occurs. It occurs at a cellular level. Hormones act on cells, and that's what controls a lot of the reproductive system. It's involved in your digestive system, your nervous system, your cardiovascular system, and so if you don't get these things, you're going to struggle later on. So it's really important that you kind of understand the importance of getting a good grounding in these processes. Okay, the third and final passive process is a little bit different from the other two. It's called osmosis. Now the definition, as you can see in the key points, and these are the key points that you need to try and get across in the test, are that osmosis is a passive process, and it is the movement of water, although it's not particularly restricted to water, from areas of high water potential to low water potential across a semi-permeable membrane in order to achieve balance. So you need those key terms in that sort of definition. Now I'm quite aware that you're not fully up on some of these terms, so it's going to take a little bit of explaining, but hopefully you can see how it works. So first of all, it's a passive process, and it is the movement of water. Now theoretically I'm going to set up a part of a membrane here, and let's sort of give it two sides of a particular container, and let's fill this container with water. Okay, so both sides of the container have an equal amount of water on. Now if we put some kind of sugar in there, let's put some sugar like glucose, is what we did in that last session, in the last slide. So this red stuff is glucose. Then if we opened glucose channels, so this glucose was able to move, then it would move. Let's draw a glucose channel just for a diagram. So draw a glucose channel there. If we open that glucose channel, then theoretically what would happen is that these red molecules would move from below to above. They would go from an area of high concentration to an area of low concentration. Now that is exactly what we described in the last slide. That would be facilitated diffusion. Facilitated diffusion. Now that is not what we're here to explain. We're here to explain osmosis. So if I draw this setup again, but this time if I put in a blue protein channel, Now the blue protein channel is a protein channel for water. So let's set it up again. We have water, and we have glucose. Now this glucose cannot use that water channel. It can't get through that channel. It's specific for water. So what we've got is this glucose is trapped in this bottom container. But water can move. It can move. And so which way would that water want to move in order to try and achieve balance? And this is the concept. Here in the top section we have something called a high water potential. It's very watery. All that there is water. Now in this bottom section what we have something called a low water potential, and that means that there's stuff in it. It's really concentrated. It's got sugar or it's got salt in there, and it's making it really sweet or salty, if you like to imagine that. And so we've got very distinct two sections, a top section which is just water and a bottom section which you can just imagine is Ribena or something very sugary or salty. Now as we can see in the key points, osmosis is about the movement of water from areas of high water potential to areas of low water potential. So you can see that water will want to move from areas of high to water potential to areas of low potential water potential. Now it's not concentration. So in the past we've talked about passive diffusion and facilitated diffusion for moving high concentration to low concentration. If we took the concentration of water from the top to the bottom, it would be exactly the same. So we're not moving water because there's a high concentration to low concentration of water. That is a common misconception or something that students write down incorrectly. What we've got there is a similar concentration of water from the top to bottom. What we've got is something called water potential, and all that means is there's stuff in it down here, but there's no stuff in it up here, and that makes it really watery or really sugary or salty. So what we get in osmosis is the movement of water from high water potential to low water potential, and what that's trying to do is trying to balance or dilute this side of the barrier, the semi-permeable membrane. It's trying to dilute this area to make it more watery, and it will continue to do so until it achieves balance, or there's no more water left to move, or there's some other force acting upon it. And that's what we mean by osmosis. It's a passive process, so there's no energy required, it just happens. It's the movement of water from areas of high water potential to low water potential. Now that means high water potential is just water, low water potential is stuff in it or diluted in it, so like, I don't know, you can imagine sugar or salt, across a semi-permeable membrane. Now that means it'll let some stuff through but not others. In this case it will let water through but it won't let glucose through in order to achieve balance. So let's set up a situation where we have a cell and we have one of these special protein channels that lets water through. Now, just for future reference, this channel is called an aquaporin. Relatively new discovery in terms of science, because we know that the body is made up of a large component of water, and inside this cell is cytoplasm, which is mainly water. But we also know, or you might know from your other cell biology lessons, is that this cell membrane is waterproof. It's made of lipids. Water can't pass it. So how does it pass it? It passes it through special protein channels called aquaporins. And if we have water, do it in big, let's get this sorted. So we have water outside the cell, we have water inside the cell. Now let's just say that they're at the same concentration. So there's the same concentration of water as there is on the inside and outside of the cell. Now consider you eating a pizza, a really stodgy, salty pizza, and I'm going to put these black dots in as salt. Salt. Now what we've created there by eating this salty pizza is an area of low water potential, because there's stuff now in it that's dissolved in it. It's made it really salty, and this now is the high water potential. Inside the cell is more watery than the outside. So what's going to happen? Osmosis, water moves from high water potential to low water potential to try and dilute that saltiness, to try and make it more watery, because a watery environment inside the body is good. It's not acidic, it's not alkali, it's kind of balanced. So what you're essentially doing there is you're dehydrating your cells. You're taking water from inside the cell and putting it outside the cell. So this becomes really flat. It becomes empty of water, and that really hinders the cell's ability to do all of its processes that it needs to do to be good at life. So that's why dehydration is not very good. I mean, it's taking water from the cell, putting it in your fluid outside your cell, when really it's inside the cell that needs the fluid, and that is osmosis. So this is the first active process that we need to explain, and as you can see, an active process requires energy from the cell, and it moves molecules not from high to low but from low to high, and it uses something called a special protein pump. Now the concept is rather simple. If we draw a cell membrane and we're going to put in something that looks like a V into that cell membrane, and all we've got there is, it's just another protein. It's a protein, and you're going to really learn the importance of different proteins. But this one, rather than being a channel, although it is a type of channel, it's a pump. It's a pump, and it needs ATP to work. It needs energy to operate and work. And what it's doing is, as it says in the key points, it's pumping molecules from areas of low concentration to areas of high concentration. Now if you spend a second identifying which side of the membrane is low to high, you should have a clear idea already. Which side is low to high? Then obviously the inside of the membrane is low, the outside of the membrane is high. So this protein pump is going to pump things from low to high. Now if this was a channel and this was open for the movement, then we'd want a natural urge for this molecule to go inside the cell, but it's not a channel, it's not letting them through. It's a special kind of protein that binds to this molecule and throws it outside using energy, because it's fighting against the natural urge of that molecule to want to come in. That's what happens naturally. So we've got to spend some energy throwing this molecule outside of the cell. Now, the same can be applied to something inside the cell. So let's say we've got a high concentration of this sort of orangey molecule, and we've got a low concentration of it outside the cell. So high inside. And this special type of protein pump, this is only one special type, actually pumps this molecule at the same time, pumps this molecule in. So we have an exchange, and this is called the sodium, Na is sodium, potassium pump. This is one special type of protein pump. One special type of protein pump, which exchanges sodium and potassium. So let me get this straight. The blue so this would be the blue would be sodium, to get my head straight. And therefore we would pump sodium out, and the orangey would be potassium. So every time we have to pump it 3 to 2, so I think it's 3 sodium out, 2 potassium in every time this thing operates. And there's a long story behind that, but you don't need to know it. All you need to know for the test is that a protein pump is active. It uses energy from the cell and it pumps things from low to high. Now we often name that against the concentration gradient. It's against the concentration gradient. So the concentration gradient, the natural wants to go from high to low, but it can't go anywhere. So we're using this pump to go from low to high, and we're using a special protein pump. And in this instance, it's the sodium potassium pump. There are other protein pumps, you know, and you're okay just to say a protein pump. That's fine. If you want to add in a bit of extra detail, you can name the sodium potassium pump, and that it pumps 3 sodium ions out to 2 potassium ions in. I hope that makes sense. So the second active process that I'd like to talk about is something called endocytosis. Endocytosis. Now the clue is in the name, in that endo means in. Endo means in, and we're going to see that across the course a few times, that endo means in. Now it's just basically the movement of a molecule inside the cell. So let's just draw a cell membrane, or a cell right there, and let's draw a molecule that wants to move inside that cell. And what it does is it moves towards the cell membrane and it butts up to the cell membrane. So if I draw that bit of cell membrane and I draw that molecule, that's where it is on that bit of cell membrane. And what actually happens is that the cell membrane starts to pinch in. So that's the molecule right there, and you can see that this molecule has sort of pushed its way into the cell. So this would be happening round about here in the cell, so it would be bulging in, and that's where the bit of molecule would be. And what happens is this molecule pinches in and is trapped inside a bubble of the cell membrane itself. So if we looked at the cell membrane as a whole, we would see that the cell, let's just try and copy the shape, something like that. Then we would see that that molecule there has ended up in its own bubble of cell membrane. So we've gone from stage 1 here, up here, to the end stage, 1, 2, 3, let's call it stage 4. And what's happened is that this molecule is pushed in on the cell membrane here, number 3, and it ends up in its own transport bubble, which is made of the same stuff as the cell membrane, because the cell membrane is just pinched off. It's made of phospholipid bilayers, and we call this transport bubble something called a vesicle. A vesicle. And that's endocytosis. To summarize, the molecule butts up to the outside of the cell membrane, it starts to push in, and it pops off, it pinches off in its own transport vesicle. Now what the importance of that is that then the cell can check that to make sure it's okay to be released into the cell. And this is how we fight a lot of bacteria and viruses. We take them into this vesicle, and if we can identify it as being foreign, or we can identify it as being bad, we can then start to chop it up and in a contained environment, so this bacteria and virus doesn't get anywhere in the cell. And again, that will be something that you maybe learn about in an infectious disease unit or further down the line. That's endocytosis. It's an active process requiring energy, and essentially the molecule pushes on the outside of the cell membrane and it pinches off in its own vesicle or transport bubble. Now, an example of a molecule that might do that would be a bacteria. So you can give that as an example in your test. Bacteria. You could also say that cholesterol does that. Cholesterol. Oh gosh, my mouse is going. Well, cholesterol. And it's as simple as that. That's endocytosis. So if endocytosis was the movement of molecules inside a cell, you may well be a clever clogs in guessing that exocytosis means getting rid of a molecule out of a cell. Exo meaning exorcism, for example, getting rid of the demons. So how does it do this? Well, if we look at the inside of a cell, and you may or may not have learned about this already, in terms of what the nucleus and the rough endoplasmic reticulum and the Golgi bodies do, but to cut a long story short, it produces molecules that are for export. So let's do this little H molecule, and when a molecule is for export, it needs to be put inside a vesicle. So the actual Golgi bodies are the packaging facility for a cell, and that packaging facility just basically wraps it in a vesicle. It's made of the same stuff as the cell membrane, and what it does is it allows it out of the factory, i.e. the cell. Now if this H molecule was produced without a vesicle and it tried to leave the cell, it just simply wouldn't get through. It would be contained within the cell and it wouldn't get out. So in order for this H molecule to get out of the cell, it needs to be put into that vesicle, into that bubble. So that bubble really then travels to the outside of the cell membrane. It's a miracle how it does that, and I'm not going to bore you with the process, but it travels to the outside of the cell. And let's zoom in, and we've got the circle or the vesicle here with the H molecule here. Now rather than sort of pushing in and buttoning out on the cell membrane, what actually happens is it fuses with the cell membrane. So this vesicle material around the outside of the H is exactly the same material as the cell membrane that it's butting up to. So what happens is this bubble fuses back into the cell membrane, in which that H molecule is able to escape. And what happens to the cell membrane is that it tightens up, so we don't get this bulge anymore, and the H is free to leave and go into the bloodstream and travel around the body. And that's exocytosis. It's as simple as that. And now a name of a molecule, now I've given it the H for a reason, because the name of a molecule that does that is any hormone that you can think of. And in fact, any protein that's shipped out, such as a neurotransmitter, for example, or a hormone. So if you can think of a hormone, I don't know, adrenaline, insulin, oxytocin. I'm struggling to think of them, although I know lots of them. It's basically how that hormone gets out of the cell. So if this cell is part of a gland, so a gland is really any group of cells that are producing hormones for the rest of the body, then you can think of any hormone that produces. So the pancreas produces insulin, for example. It's got a store of these vesicles filled with this H hormone, insulin, and as soon as that cell is triggered, then all these vesicles get released by exocytosis. I hope that's clear. So we've come to the end of the information needed to sort of nail that first question in the test. Now, I'm sure that everything may well have not stuck perfectly, so you can go back through the presentation or the video and go through it again and again. But in order to answer the first question, what you really need to know or really need to explain is the difference between active and passive transport. The difference between active and passive transport. And simply put, the active transport uses energy, passive does not. And that's it. That's it. That is the difference between active and passive transport. Now, there are 3 different active transport methods. You need to identify them, you need to draw the diagram, and you need to provide the key points for each. There are 3 passive transport processes. You need to produce a diagram for each, you need to produce the key points, and you need to give me an example for each. And you will nail the first question. Okay, so go through them, use the Quizlet, use the different resources on Moodle or on the one classroom or the class notebook, and just basically get it into your head, because you're going to have to, explain this during a test. Good luck.